Hello everyone, welcome back to Studious students. Today we'll understand the complete pathogenesis of cervical cancer from HPV infection all the way to invasive carcinoma and staging. And with that, let's begin. Why is cervical cancer so important? Cervical cancer is the fourth common cancer in women. In 2022 itself, this amounted to over 662,000 new cases and 348,000 deaths worldwide. The good news is that cervical cancer develops slowly through identifiable precancerous stages, giving us a chance to detect and treat it early. So, how does this cancer actually begin? Everything starts with infection by the human papilloma virus or HPV. Normally, the cervical squamous epithelium has multiple layers. At the bottom lies the basal layer, which contains actively dividing cells. When minor epithelial damage occurs, HPV gains access to these basal cells. Once inside the basal cells, the virus begins to multiply. After entering the basal cells, HPV DNA exists in an episomal form and starts replicating. As infective cells move upward through the epithelial layers, viral DNA replication continues. In the superficial layers, viral particles are assembled using L1 and L2 proteins. At this stage, the infection is still productive, meaning complete viral particles are being formed. But the real trouble begins because of certain viral proteins. The HPV genome produces several proteins. E1 and E2 regulate viral replication and transcription. E4 helps in viral maturation. The most important proteins are E6 and E7. E6 promotes degradation of P53, which is known as the guardian of the genome. E7 inactivates the retinoblastoma protein or RB, which normally controls cell cycle progression. Together, E6 and E7 remove important cellular brakes on growth. So, what happens if the virus loses regulation? In persistent high risk HPV infection, the viral DNA may integrate into the host genome. During integration, the E2 gene is often disrupted. Normally, E2 suppresses E6 and E7 expression. When E2 is lost, E6 and E7 become overexpressed. This causes excessive degradation of P53 and strong inactivation of RB. As a result, cells begin proliferating uncontrollably, leading to cervical intraepithelial neoplasia. These infective cells also develop a characteristic appearance. The hallmark microscopic feature of HPV infection is the koilocytes. Koilocytes show enlarged hyperchromatic nuclei surrounded by clear perinuclear halo. Whenever you see koilocytes in histology, think of HPV infection. Now let's discuss the precancerous lesions produced by HPV. Cervical squamous intraepithelial lesions are precancerous changes confined to the cervical epithelium. They remain above the basement membrane, therefore they are called as intraepithelial lesions. They are classified into two major categories. The current classification includes LSIL and HSIL. The full forms are low-grade squamous intraepithelial lesion and high-grade squamous intraepithelial lesion. Low-grade usually represents productive HPV infection and affects only the lower one-third of the epithelium. Most low-grade lesions regress spontaneously. High-grade, on the other hand, is associated with persistent high-risk HPV infection. The abnormal cells extend into the upper layers of the epithelium and have a much higher risk of progressing to cancer. This diagram makes the difference easy to remember. In LSIL, abnormal cells occupy only the lower one-third of the epithelial thickness, whereas in HSIL, atypical cells extend much higher and may involve almost the full thickness of the epithelium. Let's see how this appears under the microscope. Here we can appreciate the normal cervical squamous epithelium. The basal, the parabasal, the intermediate, and the superficial layers are arranged normally. The nucleus to cytoplasm ratio is maintained, and cellular maturation occurs as cells move upward. In LSIL, koilocytes are commonly present. The atypical changes are mainly restricted to the lower one third of the epithelium. Maturation of upper layers is still preserved. Whereas in HSIL, atypical cells extend into the upper epithelial layers. Cell maturation is lost, nuclei become larger, darker, and more abnormal. This lesion has a significantly higher risk of progression to invasive carcinoma. So, when does it become actual cancer? The turning point occurs when malignant cells breach the basement membrane. Once abnormal squamous cells invade the underlying cervical stroma, the lesion is no longer intraepithelial. It is now called invasive squamous cell carcinoma. At this stage, the tumor gains the ability to spread locally and eventually metastasize. Finally, let's quickly review the stages. Stage 0 refers to HSIL. It does not cross basement membrane. In stage 1, cancer is confined to the cervix. Stage 1 has four types. 1A refers to preclinical carcinoma. 1A1 shows stromal invasion of 3 millimeters or less. 1A2, it is no more deeper than 5 millimeters. 1B, it is greater than 1A2 but confined to cervix. In stage 2, it extends beyond the cervix and involves vagina. In stage 3, it extends to the pelvic wall. In stage 4, the cancer metastasizes. That brings us to the end of the cervical cancer pathogenesis and staging. If you found this video helpful, please like, share, and subscribe to Studio Students for more MBBS content. Thank you for watching, and I'll see you in the next video.